So hello, good afternoon. My name is Daniel Zhang.
And my name is Alexander Tiger. We represent Northern Secondary School from the Greater Toronto Area, and we're very grateful to be able to present to you our research this afternoon.
So chronic depression, anxiety, Parkinson's disease, the list really goes on and on of dopamine receptor-mediated neurological disorders. These disorders play such a prevalent role in society today that I'm sure that each and every one of you knows somebody close who is stricken by one of these diseases. So you would think that because of their prevalence, that all the funding and efforts in research that goes towards funding treatments would be very efficient in not only helping us understand the function of the receptor, but also effectively determining drug treatments. However, unfortunately, this is not the case, because in fact, we know very little about how the receptors actually work. And in fact, 90% of drugs fail in the process of research and development for these neurological disorders. So it's obvious that we need improved methods, and today we're here to provide you with one. So what we've done is we've taken the D2 dopamine receptor and we've assayed its response to various psychoactive drugs, specifically serotonin uptake inhibitors, which are common antidepressants, and alpha-2 adrenoceptor drugs, which are common antihypertensives. We then went on to quantify its off-target effects or its side effects in terms of essential individual genes, but also entire groups of genes that encode for certain parts of cell function. And how we've done this is we've transformed the receptor into Saccharomyces cerevisiae, or baker's yeast. Why baker's yeast, you may ask? Well, because of two reasons that act as essential qualities to our experiment. The first of which is efficiency, and the other is comparability. So efficiency, because you'll soon see that we managed to assay dozens and dozens of drugs upon thousands and thousands of genes in a very short period of time. But also comparability, because we know that of the 4,300 genes found in the yeast genome, 70% have human homologs, making yeast the perfect model organism for testing human receptors. Lastly, comparability, because as opposed to clinical trials, which are fundamentally very qualitative, we have developed a quantitative way of testing not only the direct effects of drugs on the receptors, but also the entire cellular response. So on to some background.
All right, so the D2 dopamine receptor is a 7-transmembrane receptor protein that is expressed on the cell membrane of neurons in the brain. It composes of 7 helices that transit the lipid bilayer, as well as an intracellular domain and an extracellular domain. The ligand-binding domain on the extracellular domain is where drugs will interact with D2. So the way we express this D2 dopamine receptor in yeast cells is we used a plasmid called the PCCW SDE plasmid, which was obtained from Dr. Igor Stagliar's lab at the University of Toronto. Essentially, the function of this plasmid is to express whatever is pasted into it onto the N-terminus of the yeast mating receptor. So when drugs interact with the D2 ligand-binding domain, the yeast mating receptor is activated, and the entire yeast mating pathway is undergone. The final 2 protein products of the yeast mating pathway code for proteins that stop the cell cycle and prepare the cell for mating. So through this method, we're able to quantitatively assay receptor activation levels by measuring the growth of a yeast culture. And we can do that very easily using a spectrophotometer. So ideally, a therapeutic drug will have a significant direct effect upon its therapeutic target with limited side effects or interactions with other proteins within the cell. However, assaying its direct effect is not the entire story. So we present to you a method of taking into account the in vivo complexity that exists within human cells and all the other thousands and thousands of proteins that exist in the cell, because the actual target of these therapeutic drugs are not present within the yeast construct that we have. So now let's take you into the lab. And the first part of our experiment was we completed a drug assay. So we transformed a strain of yeast with the D2 expressing plasmid, and that became our experimental strain for our experiment. As well, we transformed an empty vector control that became our control strain for the experiment. We then performed immunofluorescence to determine that the D2 receptor was indeed being expressed in our experimental strain. And as you can see from the fluorescence of the secondary antibody, it is being expressed where we expected it to be on the plasma membrane of the yeast cells.
So once we had our strains of yeast and we had our psychoactive drugs, the first thing we obviously wanted to do was treat the yeast with the drugs. And so on a 96-well plate, we grew up cultures of our empty vector and our D2 expressing vector in 200 micromolar concentrations of our 58 psychoactive drugs. We then ran growth curves of both these plates. And so It's important to emphasize at this point that when the drug binds to the receptor, it undergoes a pathway telling it to stop dividing, and thus we can see it evident as a growth delay on the growth curves. And so you can see on these slides, the red line acts as the control and the black line acts as the experimental for that specific well. So in doing so, we can identify drugs that are most effective and least effective in activating this receptor, to the point where if we wanted to create rank lists, we can simply integrate the curves and subtract the control from the experimental. And so, for example, in our analysis, we managed to identify MPP hydrochloride as a significant agonist. So there's a significant growth delay in the growth curve. Thus, the receptor is significantly being activated. So that's the purple line across, which is... So after we managed to identify the drugs that were most effective in activating the receptor, the next step was to quantify its off-target effects or side effects. So through a process known as synthetic genetic array, we mated our D2 expressing strains and our empty vector strains with a yeast knockout collection.
So the yeast knockout collection is a novel technique in yeast genetics. And essentially what it is, it's 4,300, it's a collection of 4,300 yeast mutants, each with one gene deleted from its genome. Another unique feature of the collection is that each strain has a unique 50 base pair barcode that allows that strain to be differentiated from all the other strains, and that'll become important in the next step of our experiment. So as Daniel mentioned, we mated our two D2 strains of yeast with the yeast knockout collection, and through the process known as SGA, which is a collection of different mating and selection steps, we were able to come up with two final sets of plates, which are basically the yeast knockout collection in duplicate. However, one set expresses the D2 receptor and one does not.
Okay, so once we had all of our 4,300 strains in the empty vector and in the D2, the next thing we wanted to do was pool all of them together so we had one culture. And, two cultures, of course, one with the empty vector and one with the D2 vector. And so from these cultures, we grew them on 48 well plates in 5 drugs that most activated the receptor as per the first part of our experiment. And this is all in preparation for microarray analysis.
All right, so once these cultures were grown to saturation, we extracted and purified genomic DNA from the entire pool. We ran PCR on those barcodes that I mentioned earlier to amplify them, and then hybridized these PCR products, or the barcodes, to the TAG4 microarray, which was specifically designed for use with this yeast knockout collection. From here, we scanned the microarrays to determine raw fluorescence values of each of the probes on the microarray. From there, we performed statistical analysis on the raw fluorescence values. For example, log2 ratios of the experimental strains over the control strains were calculated. So here's an example of a scatter plot for one of the drug treatments. You can see all the different genes and their respective log2 ratios graphed on the scatter plot. So genes that have a high log2 ratio manifested a significant growth defect when treated, in the presence of the D2 receptor. So these genes can be taken as off-target or side effects to these drug treatments. So individual genes are significant. However, what's more useful for researchers is to determine entire categories of genes that are involved in the cellular response to these drug treatments. So we then performed gene set enrichment analysis that allowed us to do this, and created what are called gene ontology, or GO maps. So when our control pool was treated with SMCPG and prazosin hydrochloride, which are examples of 2 drugs that we identified from our drug array, we find categories that have to do with mitotic progression and cell morphogenesis. And these are very non-specific, very generic GO categories that come up a lot as a cellular response to drugs. So they acted as a sort of positive control for our experiment, as we didn't expect any specific categories to come up for our control pool. When our D2 pool was treated with SMCPG, we find the category cellular response to nutrient come up in our results. Upon further analysis of the genes that comprise this category, we find many that code for amine-based receptors. So we can see that SMCPG not only interacts with D2, as suggested by our drug array, but it also interacts and has a side effect with the other amine-based receptors that are expressed on the cell membrane. When our D2 pool is treated with prazosin hydrochloride, we see one very specific category, phosphatidylinositol biosynthesis. And when we got these results back, we had absolutely no idea what this meant or what even phosphatidylinositols were. But phosphatidylinositols are a class of lipids that have a polar head on them, and they're part of the composition of the cell membrane. They had been previously identified in research as playing some role in cell signaling, but never before have they been identified as a side effect of a drug treatment such as prazosin hydrochloride. So as a future extension of our project, we would like to investigate this interaction between prazosin and phosphatidylinositol to better categorize it.
So in summary, what we've done is we've managed to quantitatively assay the effects of individual drugs on activating the D2 receptor and in essence find agonists and antagonists. But then we went on to identify, to assay the cell response to the drug treatment in terms of essential individual genes, but also entire groups of genes that are coded for certain parts of cell function. And so obviously this has many applications, the first of which is in neuroscience and drug testing. So using this method, again, we can quantitatively determine the effects of drugs on receptor activation. The most important thing to emphasize at this point is that it's comparable between drugs. You can see which drug is more effective in activating the receptor compared to other ones. So in identifying the psychoactive drugs that are most effective in activating the receptor, what this does is it sets up the platform for finding structural commonalities between the drugs to the point where we can tailor drugs specific to activating the D2 dopamine receptor, something that has never been done before. But above this, if we take a step back and take this as proof of concept of a novel new method, we have developed a way for the D2 receptor of determining, quantitatively determining not only the direct response in terms of the receptor, but also quantifying the side effects of the drug itself. But if you take any human receptor and you put it into our construct and run through the same process, you can do the same thing for not only better receptor characterization, but the streamlining of the development of treatments for receptor-mediated diseases.
So we'd like to take a moment and thank our mentors, Dr. Corey Nislo and Dr. Marinella Gabia at the Center for Cellular and Biomolecular Research at the University of Toronto. We'd also like to thank Dr. Daniel Gauci, who is our teacher advisor at Northern Secondary School. We'd like to thank Sanofi for providing us with this opportunity and funding this entire competition, and all of you for listening to our presentation today. Thank you very much.
Thank you.
We'd like to take any questions that you may have at this point.
Thank you. Thank you. Exciting stuff that you guys are doing. What do you do in your spare time?
Lots of things. Like I work for a nonprofit organization called Science Expo, and then the goal of that is to promote Science Expo.
Okay.
So the goal of that is to promote science, technology, engineering, math opportunities to high school youth, and so this has really helped embed science inside me, if you will.
Apart from science, I'm head of, or for this year, I was head of the Smart Risk organization at my school, and essentially what that is it's a organization that promotes injury prevention among teens. But in terms of other activities, I enjoy competitive swimming. In fact, I was in Ottawa for last year for a meet here. So, yeah.
Thank you. Questions?
So can you tell us a little bit about the genesis of this? Did you have an interest in looking at psychoactive drugs and then sought out a mentor where you could test some of your ideas, or did you find the mentor first and this was suggested and then you took off with it?
This whole process started out last year because we did a project last year based for Sanofi based on optimizing ethanol production and baker's yeast. And so the only commonality between those was the methodology itself, which a synthetic genetic array that we used. And so last year's project was based on optimizing ethanol production for biofuels. So we sort of wanted to continue with that at first. So our idea was finding genes essential for ethanol tolerance and then finding an ethanol-based receptor. So when in high concentrations of ethanol, it would express ethanol tolerance genes for the better creation of biofuels. However, we quickly realized that there is no ethanol receptor, and so we found a very close one for the dopamine receptor because we found that ethanol activates the dopamine receptor as well. And so one thing led to another. We realized that this could have applications outside of our project ideas from last year, and we sort of just took it from there and the snowball rolled.
Can I continue briefly? Now, some of this work, the SGAs and all of the robotics that's involved is really quite intricate technology. Can you give us a sense of how much of that you were able to do? And then in terms of the data analysis, the statistics of the gene set, GSEA, and things like that, was that all? Were you able to analyze the data yourself? And what about the the crosses and things, which are all robotically done, were these done in the lab, or did you have the opportunity to use some of the equipment?
Yeah, absolutely. So since we did this project last year in the same lab that used SGA, we were, specifically for SGA, we were very familiar with the robot that was used and the media preparation for each of the different selection steps. So we were able to basically perform all the SGA steps on our own. There was someone in the lab watching over us to make sure that, we weren't like doing anything wrong with the robot. But essentially, we completed all those steps ourselves. As well, for the microarrays, the supervisor in the lab, Dr. Marinella Gabia, she was running a parallel set of arrays for another experiment that she was working on. And so she was able to demonstrate the techniques required for microarrays. And so she would demonstrate them to us, and then we would perform them on our own set of arrays.
Alan, you of course, one of the country's experts, world experts in this whole area.
I'm curious about, obviously now you're using the pheromone receptor and the coupling mechanisms of that receptor. But obviously, the dopamine receptor would be coupled to a number of things in a mammalian cell. It would be coupled to a variety of second messenger systems. It would internalize. Does it internalize the? Probably not in the yeast, I would think.
Not in our yeast, but because the dopamine pathway is not present in yeast, it allows us to determine the off-target other effects that we wouldn't be able to otherwise.
So that part I get, but in terms of using this as a method to look at drug efficacy, what would be the advantages, as compared to, I don't know, using internalization tests or calcium or binding studies, the usual way that you would test the efficacy of drugs in a mammalian system?
I think one of the things that we really like about this project is the idea of isolating just the ligand binding domain itself. So to actually be able to quantify the receptor activation just simply through growth curves and actually do, again, I guess, so when the drug binds to the receptor, we can possibly get a numeric result, and this is essentially something that is an incredibly useful tool because, again, emphasizing the comparability of this, I think, is the most important characteristic because through a construct, if you want to determine just purely the receptor activation of drugs alone, this is very likely the best way to do so, if that answers your question.
If I could just add something to that, the important thing about using yeast is that, especially for the genetic analysis using the yeast knockout collection, nothing like this exists in such murine models, and this testing would not be ethical at all in human models. So it allows researchers and developers, pharmaceutical companies in R&D stages to not only test what effect it has on what effect that drug has on their target, but also what other effects it has in the cell before preclinical trial stages, such as murine models or even human stages later on. So it allows them to fine-tune their drugs before spending all the capital on developing the drugs on later stages.
So you effectively have an in vitro method for evaluating drugs against receptor-mediated disease. So have you taken out is there intellectual property here? Have you thought about that?
Well, in terms of the SGA, in terms of like the processes themselves, SGA has already been written up in many different papers. It was actually developed in the lab a floor above us. In terms of this entire process as a whole, we're definitely looking into possibly like the viability of getting possibly a patent on the entire process. But yeah, we're still looking into that as well and seeing if it's feasible.
I think it's really interesting because this whole process was playing around with a lot of ideas, tearing them apart, and like using steps that are most useful for us. So like hypothetically, yes, if we took this entire process of like going through this way for genetic analysis and going through this way for like receptor analysis, taken as a whole, it would be a very useful product and possibly very much patentable.
Have you talked to your mentors about this?
We've been alluding to it for a while, but I suppose once we have time to finally not work long hours in the lab, we'll have a thorough discussion of the next steps.
Absolutely.
And the mentor's reaction was?
Well, first of all, we'd asked him if this would be something that would be able to be published in a paper, and he was like, his reaction was, absolutely. What you've done here, taking elements of different procedures that have been developed and putting them all into this this method of in vitro analysis of drug effects, absolutely it's publishable. And in terms of but in terms of getting the patent, we haven't discussed that at length with our mentor, but it's something that we look forward to doing in the near future.
John?
The moment you start showcasing it at awards like this, you lose that patentability, which is an unfortunate circumstance. Do you see this technique being used by our regulators, by our drug regulators? And how would you see them using it?
So definitely we would see this method being used, because I think, again, the main emphasis on this is that we can begin to identify the off-target or side effects of drugs much before later stages in drug trials, right? And so this will essentially act as a cost saver, in the sense that if we can identify it early, then the research and development will fail at an earlier stage, but that saves us the entire process and the entire length of the entire process, essentially. And so, in addition, the direct test, just based on receptor activation, we think that in itself can act as a very useful tool for simply testing the receptor activation of various drugs and comparing which ones will be more quantitative.
And also for drugs that are already on the market today or have just been newly released, we find that there are harmful side effects that could be that are associated with these drugs, and performing an analysis like this would allow researchers and people from the drug board to see in depth which parts of the cell and which processes are actually being affected by that drug.
Any idea why the large pharmaceuticals might not have thought through this?
I suppose it goes back to working long hours in the lab, not having to think of next steps, but just trying to get the research done, get the ideas together. So unfortunately, we haven't really taken a hard, long look at who we want to target in terms of reaching the market and bringing this to the business world, if you will.
Ron?
At the beginning, you talked about one of the advantages of using yeast is that there are a number of orthologs of human genes in yeast. Well, it turns out that there are a number of sequenced unicellular eukaryotes that, in fact, the percentage of genes in those unicells are higher than yeast in terms of human or mammalian orthologs. So can you tell us what probably the biggest advantage of using yeast for this kind of work is? I think you're right that is a positive, but it's not a unique positive. So what would you say is the biggest advantage of using yeast that you can tell us?
Absolutely. So as we mentioned before, the yeast knockout collection is something that's unique to yeast, and the fact that all these 4,300 genes have been systematically knocked out from the yeast genome is barcoded for the use in experiments like these. That's something that's unique to yeast, but also the fact that yeast has this haploid-diploid duality and allows us to essentially mate two different qualities of yeast and combine them together in one double mutant in the process of SGA is something that is very important as well.
Alice, the last question.
I was interested. You did 58 drugs.
Yes.
And were they already drugs that are on the market? So did you get any interesting results from that screening where they're predicted to be doing one thing, but they're actually not activating the receptor, etc.?
Absolutely. So we actually looked into getting drugs that were on the market in order to test, but these were beyond our budget, and in fact, they wouldn't. Pharmaceutical companies would not release drugs for testing for this purpose. But we were able to obtain kits from Tokers Bioscience, and they produce drugs that have been produced that have been in R&D development and tossed aside. And so these drugs were drugs that had never been fully developed, and they sell them as kits such as serotonin uptake inhibitors and alpha-2 adrenoceptors. So we were able to get those in for the purpose of our experiment.
And so essentially this acts as a really good like a sort of molding model just to use drugs that have been in the R&D process to test our method in that process. And so essentially, I think Alex mentioned the result of prazosin, if you want to reemphasize, or just because we found this new side effect of prazosin that has presence hydrochloride that has never been identified before. And so that again is just reemphasis on how this method can identify side effects in itself, so phosphatidylinosyl biosynthesis. And so that again is just something that we would explore more in depth once we've identified the side effect. If that answers.
Gentlemen, not only are you great scientists, but you're also all-rounded individuals, swimming and doing all
Thank you
that
very
stuff.
much.
Keep up the good work. Thank you very much.
Thank you so
you.
much.